Well, here we're back for part 2 of chapter 8 on microbial genetics. Here's an interesting illustration of a change in epigenetic control of genes in which this microorganism growing in this interesting colony format suddenly switches to a completely different way of moving out into the plate. So quite a stark and interesting observation about epigenetics. We were talking about the end of last time, or at one point during our last meeting. um, a relatively recent discovery in microbial genetics and in cellular genetics in general is the idea of microRNAs. Um, some RNAs function as enzymes, you saw earlier, ribozymes. Well, here's some RNAs that function to control gene expression. And these microRNAs have some homologous sequence to a messenger RNA that it's going to influence. And when it hybridizes that messenger RNA, it's going to trigger degradation of it. So this microRNA will inhibit the expression of the gene that this mRNA was transcribed from. So you can work through this little word puzzle to see how you're doing. Mutations, changes in the genetic sequence as opposed to just changes in gene expression by epigenetics. So a mutagen is a chemical that increases the frequency of mutations. Mutations occur in the genome of pretty much all organisms at a certain rate, depending on the type of repair mechanisms they have, but mutagens increase that rate. Spontaneous mutations occur at a certain rate, but there's different things that are going to go wrong in terms of types of mutations. A base substitution is one in which there's one change in a nucleotide base in the genome at the DNA level. That's called a point mutation. So here's an illustration of a point mutation. We have during DNA replication, a thymine is accidentally incorporated in place of a opposite guanine in place of a cytosine, and then when that strand is replicated, an adenine will be put in there instead of a guanine. Now we have a permanent change in the genomic sequence of all further replications and offspring. So, in some cases, that would lead to a change in the amino acid sequence. Sometimes it wouldn't, but in this case, changing that cytosine to an adenine did change the amino acid that was called for by that codon from a cysteine to a tyrosine. So now we have a base substitution that led to a mutation, whereas in this case, the normal result would have been a cysteine and still a cysteine in that second position. All right, types of point mutations. What's their result? When that amino acid sequence does change, that's called a missense mutation that we just saw over here on the left. So again, if we have a substitution, and here's the original sequence, and then here's the altered sequence with a mutation, it's called a missense mutation. We'll have a different amino acid placed in the sequence. How about a nonsense mutation? In a nonsense mutation, we're going to have a stop codon that's going to appear where there wasn't one before. So here's our normal sequence right here. Two codons, coding for amino acids. And then here's the normal stop codon. UAA causes the ribosome to fall off and stop translating. Here we have a mutation that's going to take place that's going to result right here. That should have been an adenine. Now we're going to have a uracil there. That's going to UAG as a stop codon. So all of a sudden, that's the end of translation, whereas there used to be a longer polypeptide chain. That's called a nonsense mutation. So missense is substituting a base. We went from a glycine here to a serine. Nonsense is inserting a stop codon, so that we're going to have a truncation, early termination of transcription and translation, I should say. A frame shift mutation. If we either insert or remove a base, instead of just swapping the identity of a base, we either insert one and change the orientation of all the codons thereafter. That's going to be a frame shift mutation. So here's our original sequence, methionine, lysine, phenylalanine, and glycine, and then a stop codon. And now we're going to lose an adenine right here. You see there's 3 adenines right here that called for a phenylalanine. We're going to lose one of those. And so now the codon reads adenine, adenine, cytosine, and that codon calls for a leucine, and then an alanine. So everything got shifted one base, so none of the codons are going to read correctly after that. That's called a frame shift mutation. It turns out that when that happens, relatively quickly, a stop codon will appear. It's pretty unusual for any length of DNA code to be absent of a stop codon unless it's a specific gene, which has been handed down through however many generations. So it's designed to have no stop codons, but if you randomize things by doing a frame shift, a stop codon will pop up relatively soon. What's the frequency of mutations? Well, spontaneous mutation rate depends on the organism, but about 1 in every 10 to the 9th base pairs that's replicated, or 1 in every 10 to the 6th genes, right? Because every 3 bases produces a codon or calls for a codon. Mutagens are chemicals that can increase that rate to from 10 to the -5 to 10 to the -3 per replicated gene. So that's it doesn't sound like much, but when you remember back to the fact that every time an E. coli divides, it's going to have to replicate 4.5 million base pairs to make a copy of its genome. And among those millions, if we have mistakes popping up every 1,000 or so, that's going to produce some potential problems. One way that mutagens can arise is by changing the chemical structure of a nucleotide base. Here's a nucleotide, and it's actually a nucleoside. I guess there's no phosphate on there. It's a ribose sugar with the base on there. A purine is attached on there, and it's supposed to have this amine group, and if it gets oxidized to an oxygen, now that'll pair with the wrong partner. Instead of pairing with thymine, it's going to pair with cytosine. And so now we have that oxidation happening. Nitric acid here is being added, oxidizes that, that base. So now it doesn't pair. This adenine no longer pairs with thymine. When it's the original strand will produce the correct match. We'll have a thymine and adenine in the double-stranded. Now this modified adenine is going to call for a cytosine as its partner. And then when that gets replicated, we'll have a guanine there. So we're going to have a perfectly a mutation taking place that way. So oxidizing agents, other chemicals can act as mutagens. Analogs. Sometimes we intentionally introduce a modified nucleotide into the environment in which some organism is growing as a medicine in order to spoil its normal transcription and translation or replication so a microorganism can be stopped in its tracks and force mutations in there. So here's an example of an analog of adenine, 2-aminopurine. See, we have we take away that amine group right here. Now we have a hydrogen there instead, and now it's going to cause mutations. So analogs are just modified nucleotides that will result in changes in the DNA sequence when they're incorporated into the DNA during replication. As we'll see later on when we talk about antibiotics of specific types, there's quite a few that are nucleotide analogs. Ionizing radiation, that can cause changes in the deoxyribonuclease, the DNA sequence. UV radiation can also cause damage. So X-rays, we said, is ionizing. That causes major bad chemical changes in the DNA. UV radiation, what does it do? It causes thymine dimers. So here's a pair of thymines that just happen to be next to each other in the sequence, and when they're exposed to enough energy of ultraviolet light, will form a chemical bond between these thymines. It's called an adduct, and some repair enzymes will come along and recognize that and cut it out and replace with the proper bases. That's the normal response to that. But sometimes that doesn't happen, and these these two thymines will end up causing a mutation to occur during replication of the DNA. So that's called nucleotide excision repair. Excision is this cutting out of the mistaken DNA and replacing it with the correct original sequence. That's why you don't get a cancer every time you get a sunburn, because you have plenty of repairs that can go on in your cells. You can either delete cells by apoptosis that are too badly damaged by ultraviolet light, or you can do this excision repair job on the genome of some of your skin cells and come out okay. All right, let's talk about selection and replica plating. Here is a pattern of some colonies growing on a particular medium containing histidine, and we're going to discover a strain, a particular genetic change in one of the colony forming units that then led to a colony that cannot synthesize histidine and therefore can't grow in its absence. So we have this plate full of colonies. Maybe a mutagen had been added to it, maybe not. We'll take this velvet, it's basically a little cylinder covered with velvet that can fit right down inside this plate. We're going to mash it right down on all those colonies, lift it up, and now it has bacteria or fungi all over it from these colonies, and then mash it down onto a brand new plate, and we should recapitulate that original plate exactly. However, if we do the same thing, we mash it down a second time onto a plate in histidine minus medium, there's no histidine present, we find that one of the colonies is not present, is no longer there, that was there. That means that we now know that this particular colony arose from a mutated cell, a mutant cell, that cannot synthesize histidine any longer. Something happened, it lost an enzyme, and it no longer can make histidine. Well, that's the basis for this idea for what's called the Ames test. Pretty cool idea. And Salmonella bacteria that have a single mutation that makes it so they can't synthesize histidine, they can't grow on histidine minus media, are used for a test of mutagens, chemical mutagens. And so we're going to take some of those mutated salmonella plus a putative mutagen, suspected mutagen, and mix it with some rat liver extract, and I'll explain in a second. And we'll mix that together and plate that out, and we wind up with a bunch of colonies that are now able to synthesize histidine. This is histidine minus media, and these salmonella were histidine dependent, and now yet they're able to grow on a histidine minus plate. Whereas just the salmonella bacteria alone, these modified salmonella plus rat liver extract, cannot grow on the His minus plate. There's a couple of spontaneous revertants, they're called, or mutations, have restored those some salmonella to being able to synthesize histidine once again, reverse that original mutation that was present. So let me see if I can clarify that one more time. In order to test a chemical for being a mutagen, enhancing the rate of mutations, we'll take some experimental bacteria that can't synthesize histidine, add the mutagen, which might then cause some mutations that restore the ability to metabolize histidine, mix with rat liver extract, and voila, get all these colonies which are revertants, meaning they're back to the old wild type capability of synthesizing histidine. They can grow on His minus plates. So yes, this suspected mutagen is a mutagen that caused all those genetic changes, and some of the cells were able to go back to the original histidine making capability. And this is just a control down here showing that it was the mutagen that did this because rat liver extract plus those experimental salmonella can't grow on His minus, except for there were a couple of colonies where a spontaneous mutation occurred that caused the experimental bacteria to revert back to their natural form in which they could grow on His minus media. That's called the Ames test for mutagens. If there's things that people might eat in their diet, for example, that might be considered dangerous because they might cause DNA mutations, we can test it with the Ames test. If a lot of mutations occur, we might say, well, you shouldn't eat those heavily grilled substances on your grill because some of those products of the grilling are mutagens and so forth. Why is the rat liver extract in there? Well, a lot of compounds that might potentially cause mutations or cancer are not mutagens in their own right, but when the liver gets a hold of them, the liver has a ton of enzymes in its cells that can form intermediate compounds from those substances that can then produce mutagens. So maybe this isn't a suspected mutagen until it gets mixed with rat liver extract, and then it can turn into a mutagen. So it's not just the things that we eat in our diet that can be environmental hazards for cancer. They can be things that the liver converts into hazards that can cause mutation and cancers. Okay, that was a pretty cool thing. Um, let's talk about horizontal gene transfer. What do you mean by that? And this is a big problem clinically. Vertical gene transfer just means that the typical thing. We have a we have a bacterium which is going to replicate. It makes a copy of its own genome, and then its daughter cells receive that. So here's the original genome, and here are the two daughter cells, two product cells, whatever you want to call it, have identical copies of that genome. Well, if a mutation occurs in this parent genome, it will then appear in the offspring in the next generation of bacteria. That's called vertical gene transfer. Horizontal gene transfer means within the same generation, bacteria can swap genetic information by several mechanisms: transformation, conjugation, transduction, and transposition. So I want you to be able to, after you study this chapter, give a fairly detailed explanation of what do we mean by transformation. How is genetic information being acquired by a cell that didn't have it before by transformation? How does a cell acquire genetic information by conjugation and so forth? So let's take a look. Here's an interesting thing that can happen: crossing over. If there's a little bit of a homologous region between DNA sequences on two different chromosomes, for example, or part of a chromosome, a DNA sequence can hybridize with the homologous part of the sequence in this recipient chromosome, and then some enzymes, or there's a recombination enzyme, REC-A it's called, that can end up swapping out part of the chromosome, so that we now have one of the copies of each of these two, the donor and recipient, have swapped the sequence, and there could be some genetic information that wasn't even present in this genome that was present in this genome, and now it's been delivered by this recombination event. So whichever bacteria have this REC-A protein, they have a high likelihood of recombination happening in their genome over the course of many generations, dividing of cells. All right, let's talk about transformation. Are you ready? Transformation means a bacterial cell taking up some naked DNA, just a piece of DNA, taken up from its environment, which then, once it's inside the bacterium, is incorporated into its own genome. For some reason, bacteria do this. So if there's some DNA floating around from a bunch of dead bacteria, for example, that got all degraded, a bacterium can easily take up a piece of that DNA, incorporate it into its own genome, and now whatever genes happen to be present on that fragment will now be part of the genome of that bacterium. So first I want to show you this really cool experiment in which this was demonstrated for the first time or discovered. Streptococcus pneumoniae is a bacteria that, when it has a gene to make a capsule, can cause pneumonia, can cause disease. If it doesn't have the gene to make the glycocalyx, make that polysaccharide capsule, it's harmless. The immune system can whip it. And so this genius scientist showed that if you injected the capsule containing strep pneumoniae, am I saying that right? Something sounds wrong about that. Anyway, if the capsule gene is present, pneumonia will occur, and the rat will die, or the mouse will die. If the gene for the capsule is not present, the mouse will not be harmed. The immune system will take care of that infection. So here's the trick, though. If the scientists found that if you heat killed the bacteria that had the capsule gene, inject them into the mouse, nothing happens. There's no live bacteria to cause an infection and cause pneumonia. However, if you take those killed bacteria that were just shown to be harmless and mix them with the harmless non-capsule gene containing strain of the bacteria and inject them into the mouse will die. Why? Because all those dead bacteria that had the gene for the capsule, pieces of DNA are floating around in the gamish with all those dead cells. At some point, one of these non-capsule producing bacteria took up the gene for the capsule production, and now we have the virulent or disease-causing bacteria in action, causing pneumonia. So that is the coolest thing, demonstrating that these non-capsule producing cells took up the gene and became able to produce capsules and killed the mouse. Before that, it was thought that always vertical gene transfer. There had to be a mutation in a bacterium that then would be handed down. That would be the only way to go. It turns out that horizontal gene transfer is a thing, and that's a bad problem because if there's a bad player in the mix, and even if it's being killed, it can transfer some virulence genes, some disease-causing genes to other bacteria. So this is just a cartoon to illustrate. You have some naked DNA, and we have a bacterium who will take up some of that naked DNA from its environment, and then that DNA may fuse with the genome, maybe a part of it, and as long as there's enough of a gene present in order to add some capability, it may change the phenotype of the cell, genotype and phenotype. All right, the next way for horizontal gene transfer, sex pilus. Some bacteria have genes that allow them to create a pilus, which is like a hollow tube connecting it with another bacterial cell. When a bacterium has the capability to make a pilus, has the appropriate genes, it's called an F plus cell. And here's a naive cell that doesn't have the ability to make a pilus. It's called an F minus cell. But now genetic material can be transferred through the pilus. So here's an example. Here's an F plus cell. It forms a pilus between it and another bacterium, and it starts to transfer this F factor. In this case, it's a plasmid. A plasmid is a small piece of circular double-stranded DNA that, as we said, bacteria commonly harbor. And in this case, it happens to have the genes for making a pilus, and it will swap a copy of that plasmid or factor right over into the F minus cell. And now the F minus cell has acquired the genes to make a pilus, so now it's become an F plus cell. So now it has acquired some new genetic information. It's a new man altogether, woman, bacterium. All right, well, sometimes the F factor can be incorporated right into the genome of the host bacterium. And now, when this cell fires up its genes for making a pilus and starts to do the transfer, it may transfer the F factor genes along with some of the genetic information that was part of that first host bacteria. So this donor bacterium, which is called a high frequency pilus and transfer bacterium, HFR cell, because it has the F factor incorporated into its own genome, it's going to transfer over the F factor information genetically, and which is going to incorporate itself into the recipient cell's genome, and along with some genetic information from that donor cell. So there's potentially a gene that's again could be a disease-causing gene. The way they drew this, this became an F minus recombinant cell, meaning it has a new gene that came from the donor. It could have actually been both at the same time. It could have become an HFR cell itself plus a gene that causes disease or causes antibiotic resistance or something like that. So a third method of horizontal gene transfer is called transduction. So we had transformation and I already forgot that term. Conjugation, sorry, I'm spacing out, I'm getting tired. And transduction. Transduction is a phage-related mechanism. Phage are viruses that infect bacteria. They're actually called bacteriophage. We call them phage for short a lot of times. So here's a phage. The way it works is that it injects its genome into the bacterium, and then the bacterium, and it produces enzymes that get translated that cut up the genome of the host cell, and then also the genome of the phage forces the production of all the proteins needed to build new phage particles. That's what viruses do. They take over the machinery of a host cell and force it to form copies of the virus. So now you have a whole bunch of new phage that can burst out and infect another cell. Well, it so happens that during this normal process of a bunch of new phage being produced, right, copies of the genome and then copies of all the proteins, some of the fragments of the host genome of the bacterium could be packaged up in a phage particle. The phage particle is just a capsule of protein coat that's around the genome, and then when that gets introduced or injected into a recipient cell, now we're not injecting phage DNA, we're injecting bacterial DNA, including a gene that maybe causes disease or a gene for antibiotic resistance or whatever it might be, can now be incorporated into the genome of that cell and then given to all its offspring. So now we have transferred genetic material from this donor cell into this recipient cell by way of a phage particle. So that's called transduction. That's just the name of the process. When a phage injects DNA into a bacterium or a virus into a eukaryotic cell, it's called transducing that cell, transduction. Specialized transduction. Here we have a phage. A prophage means the phage genome has already been incorporated into the genome of the host cell, right? In this case, the phage genome was never incorporated into the host cell. It was just in there, got replicated, and then was used to transcribe and translate some proteins, make new phage. Here, the prophage is the phage genome incorporated right into the bacterial gene, chromosome. And so then when we package up a bunch of phage particles, they can contain not just the phage genome, but also a gene possibly from the genome of the bacterium. So here we see this phage that has like a hybrid that it's delivering. It's delivering the phage genome as well as a gene from the original host bacteria, the donor bacteria. When that then fuses with or inserts itself into the bacterial chromosome, we've introduced not only a phage, so we not only reproduce the prophage, we have a gene that came from the other bacterium. Again, that could, in this case, they're using it as an example. Galactosidase gene, I think, is what they're talking about. But it could be anything. It could be a bacterial disease-causing virulence gene. It could be any number of things. It could be an antibiotic resistance marker. So those are multiple ways that horizontal gene transfer can occur. So make sure you can run through this list of mechanisms of horizontal gene transfer and explain what's going on there. How can genetic material from one bacterium be acquired by another bacterium that has nothing to do with any cell ever dividing? It's just being transferred over by one mechanism or another. Plasmids are, again, those small circular pieces of double-stranded DNA that are harbored by a lot of bacteria, and sometimes they have antibiotic resistance genes. They might have genes that can cause disease, like a capsule-producing gene we talked about. They might have the genes for making a pilus, the F factor, or R factors are plasmids that contain antibiotic resistance genes. Antibiotics are supposed to kill bacteria and poison them, but some bacteria have a gene that allows them to destroy the antibiotic or pump it out of their cells or whatever it takes to be immune to that particular antibiotic. So an R factor is a plasmid that acts in that way. So here's a picture of a multi-resistant R factor. All these different three-letter titles refer to genes that produce products that can allow bacteria to be resistant to different types of antibiotics. Tetracycline, for example, is a resistance marker for tetracycline. So, troublesome. One bacterium becomes resistant to an antibiotic, it can spread in the whole bacterial community within the body, cause all kinds of problems. Transposition, I guess we didn't even make it through our list of 4 things, did we? Transposition was the fourth one, and we're just now coming to transposition. What in the world is a transposon? Transposition. So a transposon is a sequence of DNA that has in it a gene that can recognize a sequence on either end of this thing called the transposon, this sequence of DNA called the transposon is like a signal sequence that the enzyme can cut it out and then splice it somewhere else in the genome of the organism or in another organism. That's what transposon is. So here's a transposon. Simplest case of a transposon, it's a signal sequence, an inverted repeat that'll be recognized by the product of this transposase gene, a protein enzyme that will be able to say, oh, I see this end and this end, cut it out, now it's free floating, and then the transposase protein can then insert it in a different location in the genome. So that's the simplest case. What if the transposase gene wasn't the only gene present in the transposon? Here's a kanamycin resistant gene, an antibiotic resistance gene that's included in here. So here we have two different transposons with those. Each one, we can see this transposition sequence on each end. Now when the transposase, which is situated here, is translated and transcribed, we turn that around, transcribed and translated, it can cut out this whole cassette and move it into a different spot, move it into a different bacterial genome, and now that bacterium has just acquired the kanamycin resistance gene. So now it's an antibiotic resistant species, whereas it wasn't before. So transposition, jumping sequences of DNA by virtue of an enzyme that's incorporated in one of its genes. So here's the transposase gene. It's going to produce a transposase. The kanamycin resistance gene is caught right in the middle of this transposon. It's going to be inserted by the enzyme right into the genome of the bacterium, and they're going to cause it now to become resistant to kanamycin, just like an antibiotic. So if you get a mutation, and that mutation confers some kind of advantage to the microorganism, now that organism is going to be selected for. So not only can horizontal transfer allow populations of bacteria to acquire some disease-causing gene or an antibiotic resistance gene, now we're going to select for it. Potentially, if it's an antibiotic resistance marker and we're poisoning bacteria in someone's body with antibiotics and some bugs become resistant to that, what's going to happen? They're going to take over and grow because they're no longer susceptible to the antibiotics. That's what we mean by selection. And now we're going to have a big problem because we're going to have mainly resistant bacteria left at the end of all that. West Nile virus, we mentioned that as an emerging pathogen that causes encephalitis. Studying genomics can help nail down the origin of some viruses. Sometimes viruses just appear in the world. All of a sudden, a whole bunch of people are getting sick, and it's being spread from person to person in different parts of the world. We don't know why. Where the world did it come from? Well, we can study the sequence of the genome of viruses and figure out which ones are more connected to or more related because they have closer matching sequences. We saw this little table earlier already, and help figure out what was the sort of evolutionary path taken by this viruses. Viruses mutate very, very quickly, and so how did we get from the beginning to where we are today? And sometimes it's discovered that a virus that's endemic in a population of animals in some part of the world acquires some mutations that allows it to have a change in host range, meaning it can now infect humans, and then we have a brand new problem, brand new disease on our hands, an emerging disease. And we can figure out where it came from by looking at the relatedness by the sequence of DNA in a particular strain of the virus with maybe a virus that's discovered in some animals in the jungle or whatever. Again, that's what we were talking about way back earlier in the chapter. So that's our story about microbial genetics. I hope you enjoyed that. It's pretty fascinating to discover that bacteria can swap genes. They can become very much disease-causing when they were perfectly benign by just getting some genes from other bacteria in the same host body. Join me soon for the next chapter, chapter 9. Hopefully that'll be coming up tomorrow.